It's an honor and privilege to welcome Dr. Amanda Lauer, who is an associate professor and the co-director of the resident research training program in otolaryngology. She comes from the Department of Otolaryngology, Head and Neck Surgery and Neuroscience at the Johns Hopkins University School of Medicine. So Dr. Lauer, thank you very much for joining us, and we're looking forward to your talk.
So I'm so happy to be able to talk to you about one of my research passions, which is the auditory efferent system. And, I'm happy to take questions during the talk if there's something that needs more clarification, or at the end. Either way is fine with me. But before I start on the data, I want to first give an acknowledgement to my postdoc, Sergio Vicencio Jimenez, who is the senior postdoc in my lab. He's the primary driver of the Olivocochlear data collection at this point in time. And he's also a great teacher, so he's helped me a lot with some of the introductory slides in this talk. I have no disclosures, no financial conflicts of interest. And I'll just quickly go over the learning objectives for today. So I just would hope that you all are able to understand something about the hypothesized roles of Olivocochlear efferent system in hearing, how this is influenced by acoustic experience and age, and about emerging research directions or areas where we don't know anything in the Olivocochlear efferent system. Always happy to help inspire new research in this field. So I think everyone has probably seen some version of this textbook central auditory pathway schematic, which makes things look really nice and streamlined and simple, going up from the ear all the way up to the auditory cortex. But I think maybe people don't typically see these reality-based diagrams of the central auditory system, which includes very complicated ascending pathways with lots of crosstalk across hemispheres and between different centers. And even some non-primary, non-auditory areas that are providing input to the auditory system or receiving input from the auditory system. And then, of course, there are descending pathways, which are really not as well understood as the ascending pathways, but probably almost equally as complicated, and going all the way from the cortex to the inner ear. And so why do we need to have so many of these descending efferent projections? It's really because perception is an active process, even in its most passive situations. We need sensory efferent feedback so that, for instance, we don't see the world move when we scan an image. We need it so that when we generate our own sounds through speaking or chewing or sneezing, they're not perceived as extremely loud and annoying or even potentially damaging to the ear. And the auditory efferent system actually is fairly sophisticated in that it consists of a whole bunch of different loops among different levels of the auditory system, top-down loops. And what I'm primarily interested in my lab is this loop between the brainstem, the superior olivary complex, cochlear nucleus, and the cochlea. So this is a very simplified version. And basically, I've just ignored all of the auditory cortex and inferior colliculus higher order input to the system at this point in time, just because it's so very poorly understood. We're going to focus here on the brainstem to the ear circuits. So this system provides ipsilateral and contralateral input to the ear from the superior olivary complex and the periolivary areas. And we have a medial and a lateral branch. The laterals are primarily projecting ipsilaterally. The medials are both crossed and uncrossed. Okay, and if you look in at the ear where these things form terminals, we primarily know about the outer hair cell efferent synapse here with the medial olivocochlear neurons. These are forming huge direct synapses with the outer hair cells. There are also these unmyelinated lateral efferents that form synapses with the type 1 spiral ganglion neurons in the vicinity of the inner hair cell, at least in adult animals and humans. These are not very well understood because they're neurochemically complex And because they're unmyelinated, they're kind of hard to study with physiological techniques in general. So most of what I'll talk about is relating to the medial efferents today. We'll throw a little bit of lateral efferent data in there. So these medial efferent synaptic mechanisms are quite well characterized. This synapse provides inhibition to the outer hair cells via a cholinergic receptor called the alpha-9, alpha-10 nicotinic acetylcholine receptor. And this is just made up of these two alpha-9 and alpha-10 subunits. What happens is you get release of acetylcholine from the medial olivocochlear synaptic terminal, and that excites these receptors, which causes a calcium influx into the outer hair cell through a cascade of events. This also results in a potassium outflux. Okay. This basically just hyperpolarizes the outer hair cell, reducing the cochlear gain. Okay. So what happens with the peripheral activity, the physiological activity, when these outer hair cells are hyperpolarized, is that you stimulate the olivocochlear system, you inhibit this gain, and now you've got an increased threshold in your auditory nerve fibers, or reduced response, I should say. And what happens is this actually changes the rate level functions of the auditory nerve fibers. So here we're looking at the discharge rate of one auditory nerve fiber across different sound pressure levels of a tone that is played in quiet or noise. So here we've got what happens with the response to this tone in noise alone, right here. You've got kind of a compressed response. Sorry, I think somebody's not muted in the background. You've got a very compressed response across different levels. So the neuron is not really able to represent change in level very well, right? When you stimulate the olivocochlear neurons, you get a reduction in that baseline response to the noise alone, and you get an increase in the saturation rate. And what this does is it really increases this change in rate representation. So this works very well for a transient tone in noise, meaning that it makes the tone sort of pop out of the noise physiologically. And what that should do is make that tone pop out of the noise as well, perceptually. However, things are never as simple as one might envision from a single auditory nerve response function, right? So there's been a lot of debate over many years about what this pathway activation actually does. What does the olivocochlear system actually do for hearing? So there are a bunch of hypothesized functions. I'll talk a little bit about three of the main ones here. One is this anti-masking effect, like I just described with a tone, meaning that you're suppressing the background noise response, which makes any sort of transient non-background noise signal pop out, right? And it makes it easier to hear in a noisy environment. Another thing is that because you're suppressing that response to the noise, this can be actually protective against chronic noise exposure, chronic acoustic trauma. And another thing that has been hypothesized is that suppression of certain frequencies that you're not paying attention to could be going on here, so that you can devote all your resources to the sound you're expecting to hear. Or conversely, if you're doing a task in another sensory modality, such as a visual task, this actually suppresses activity in the ear, allowing for selective attention to the visual task. So I'll just tell you now, there are data for and against all of these hypotheses in the literature. Primarily, the literature has focused on anti-masking and the protection from acoustic trauma, and has shown effects under certain conditions in those that are perhaps a bit more obvious than the selective attention effects. But there is some indication that under the right stimulus conditions, all three of these functions can happen with olivocochlear stimulation. All right, so why is it complicated to really definitively say this is what the olivocochlear system does? Well, we're not just dealing with that peripheral synapse that I just explained in kind of a simple manner. The system is actually pretty complicated once you work in all of the central pathways. And there are a lot of areas where there could be plasticity in these pathways or dysfunction. So this would include the primary interneurons in the cochlear nucleus. There are some axon collaterals from the olivocochlear neurons to cochlear nucleus. And the system gets input from a lot of other sources, including inhibitory inputs from the superior olive, auditory cortex, inferior colliculus inputs, and also neuromodulatory pathways, so serotonin, dopamine, norepinephrine, things like that. So it becomes kind of complicated, even if we assume that once the system is developed and in place, it's static. But of course, we know nothing in the nervous system is ever truly static. And it's most likely highly influenced by an individual's acoustic exposure history or their acoustic experience history. Unfortunately, as we all know, people are very, very uncontrolled in this respect. And some people are, exposed to very loud sounds. Some people are practicing music, which may give them specializations in listening to certain sounds while at the same time being exposed to loud sounds. Some people are using ear protection while they're doing this, and some are not. And another complication of acoustic exposure and acoustic experience is that a lifetime of listening to all that loud music and your motorcycle and your gardening equipment will accelerate age-related hearing loss. However, yet another complication is that many people who have hearing loss will be wearing hearing aids. And so this is not to say that's a bad thing, that's a good thing for healthcare, but it is yet another complication in trying to understand the role of the olivocochlear system because it's going to be influenced by all of this acoustic experience, in addition to things like earplugs to protect against hearing loss. All right, so we primarily use animal models to get at what this system is doing. And we like using mouse models because they're convenient and because they age in a pretty short time frame. 3 years is old for a mouse. And we can use different models that have sort of a slow time course of age-related hearing loss development, or a short time course. We can use genetic mutants as well. And so we've leveraged the fact that there are several mouse models of medial olivocochlear dysfunction out there. The primary one being what we call the alpha 9 knockout mouse. And in this case, it's just missing the receptor subunit, the alpha 9 receptor subunit at the medial olivocochlear synapse. And basically what this does is it just it disrupts the outer hair cell hyperpolarization and eliminates some of those classic cochlear gain effects. So here we're seeing a compound action potential from the auditory nerve in response to increasing tone levels. And normally what happens when you shock that olivocochlear axon bundle with an electrical shock, this will shift this function over so that the system is less responsive to any individual sound level here. And you see in the alpha 9 knockout mouse, that shift, that inhibition is gone, that suppression is gone. So they're sort of functionally de-efferented. I'll tell you the synapse is still physically present, it's just not working the way it should. And the hypothesis with these animals a long time ago, put forward by my postdoctoral mentor, Brad May, and his collaborator, Cynthia Prosen, was that these mice would have abnormal hearing and noise behavior, and that this would be the first animal study to really definitively show that. Previous animal studies have provided equivocal results. So what they did was they trained mice to hear a tone in noise, shown here in the critical ratio plot, or to detect a change in the intensity in noise. And the hypothesis was that both of those behaviors would be impaired in the knockout mice. But as you can see, there was no difference between the knockouts and the wild types in these two tasks. And this was a bit of a surprise based on what the hypothesized role of the olivocochlear system is for aiding hearing and noise. However, something interesting actually happened, and that is that there seemed to be a bit of a worsening of detection of the tone and noise with age in the alpha 9 knockouts only, and not in the wild type mice. And this really didn't go anywhere for a while until I started working with Brad May, and we started really looking into this more closely, thinking maybe what's going on is these mice are daily being tested in noise. And so they're getting exposed to noise for months while these data points are being collected. And so maybe that daily exposure to the background noise is actually causing some impairments in the alpha 9 mice. And so I followed this up, asking the question, does chronic exposure to this environmental noise worsen hearing when you have olivocochlear efferent dysfunction? So what we decided to do at that point was not do a trained behavioral task as in the previous study, because what can happen with animals who are doing a behavioral task is, they're listening to the same sounds for many trials every day, and they're getting rewarded for it. So there may be some sort of compensatory mechanism that takes over, allowing them to improve their performance, right? Improve their hearing and noise that we're not tapping into when they are doing a highly practice routine task. So we use the auditory brainstem response measurement, very common measurement of hearing in animal models because of its efficiency. It's basically just putting 3 subdermal needle electrodes on the scalp of the animal, and well, I should say 2 on the scalp and 1 in the hind leg. And we just play sounds and average the response. This is very similar to the clinical ABR, except that we get a bigger signal to noise than what you would get in a human because of the little size of the mouse head and the lack of substantial cortical tissue generating responses compared to humans. So this is a typical mouse ABR, and you can see it's very peaky. We get at least 4 to 5 peaks, fairly simple to measure. In an anesthetized mouse, in about 40 minutes, you can get a whole audiogram. Turns out that Johns Hopkins gave us a very nice manipulation for different acoustic exposure conditions for housing these mice, for aging these mice. And basically what happens is the rooms where there are a lot of animals housed and a lot of experimenters going in and out, and these ventilated cage changing stations, these micro barrier stations cause a lot of noise for the animals. And this really happens during the workday and mostly goes away during the weekend. So we know that it's anthropogenic noise primarily, not from the animals themselves. If you look at the noise in the region of the frequency regions where the mice can hear, really it's more on the low end of their hearing, not so much in the ultrasonic range, but you can see quite a lot of variable noise happening during the day. So this is like the mouse's work day, right? The mouse going to lunch, really it's the people going to lunch, but this is the type of exposure it's mimicking. And then in a room where there are not very many different investigators housing mice, there's not a lot of equipment, there's no individually ventilated caging systems, you get much less of this noise throughout the day. So mice are living in this, they're exposed to this every day, and we track the hearing with time for about a year and a half. And basically what we hypothesized was that the alpha 9 knockout mice would show early age-related hearing loss. However, we found something really surprising, and that is that even in early adulthood, which is about 1 to 5 months here for a mouse, the thresholds were mostly normal, except some of the alpha 9s showed slightly higher low frequency hearing at 8 kilohertz when housed in noise. So 8 kilohertz low for a mouse. However, there were no differences in the aging animals at all. So noise didn't seem to have an effect in the wild types or the knockout mice, and the knockout mice were not different from the wild types in any of these conditions. So it was a little bit disappointing. And And at the time we decided to shelve the project. This was sort of probably almost 10 years before I actually published the data in 2017. And it just was at a time when you really couldn't publish negative effects. So we moved on to other things. And in the interim, this story of cochlear synaptopathy really started emerging, and looking at ABR wave amplitudes as a proxy measurement for cochlear synapse loss started gaining popularity. We started also noticing some other effects coming out of the literature, such as a study from Charlie Lieberman showing that if you did a lesion with the olivocochlear neurons, either a surgical cut to the olivocochlear bundle for the medial efferents, or a neurotoxic lesion to kill the lateral efferents, these animals actually did show accelerated age-related hearing loss just when sitting around in their housing environment. We also communicated with some colleagues in Argentina who were doing some aging studies with a gain of function and mutation. So this is where the alpha-9 receptor subunit actually has increased function. And they were showing, even in a year, not really super old for an animal, that there was protection against age-related hearing loss. However, these animals actually have already elevated age-related hearing loss. So elevated thresholds, sorry, not age-related hearing loss. So there just really isn't much of a shift when the thresholds are already elevated. We re-examined our data by going back and looking at the wave 1 amplitudes to try to get at some sort of subclinical type of damage. And we did see actually here some differences, primarily in the noise groups. So here we noticed that ABR wave 1, which is coming from the spiral ganglion neurons, was lower in the knockout mice housed in noise compared to the wild type mice housed in noise. And this was true for a broadband click and for higher frequency sounds. At least it was significant for those, but you can see the effect is really there for all the frequencies we tested. However, if you look really closely at the data, it's not so much that the knockout mice in noise have lower thresholds than the other groups, but rather that the wild types housed in noise had, better wave 1 amplitudes than the other groups. And so we hypothesized this might actually be a protective effect of being housed in this noise, which itself was not so much damaging. And this could be because of excitation of the olivocochlear neurons actually providing protection. So our next question was, does chronic exposure to environmental noise alter the olivocochlear efferents? And the short answer is yes. We did a more controlled noise here, a short-term 1 week 85 dB broadband noise, and labeled up the olivocochlear synapses in the cochlea using SV2 stain. And basically what we see is that there's not much of an effect in those medial efferents, but there was some upregulation of these lateral efferents. We had more staining, more SV2 staining you can see it here, in the noise housed animals. Now, I wouldn't recommend everybody spending a week in 85 dB noise, because this will obviously cause some damage accrual over time, but there's more of a proof of concept here. We decided, rather than trying to deal with long-term these potentially damaging noises, we switched to what happens to the olivocochlear system with natural aging. So instead of asking, how does it protect against natural aging, we're asking, how does it change with natural aging? And the reason for really looking at natural aging is that in the human literature, it's really unclear what happens to the olivocochlear system with age. So there were a few earlier qualitative studies showing that either there is partial loss of these efferent neurons or there's no apparent loss of these neurons. Again, completely conflicting data. And really, we aren't even sure what normal is in humans. Here is some synaptophysin labeling of these terminals in young humans' ears, basically showing an apex to base gradient in the labeling of the medial efferents. There's almost nothing labeled here, the apex for these medials, and there's increasing labeling in the base. And there's a little bit of data from very large age groups of animals showing that this pattern changes with age as well. This is across cochlear frequency regions for the medials and laterals. Primarily, these changes are in the higher frequency regions for the medials with age, indicating some loss. But the young group is 0 to 40 years, which is a really large range. 20 to 40 years old is not necessarily normal. And I think we all know how hard it is to get hold of really good quality human temporal bone tissue from younger donors to really get at this question. So again, we turn to our mice. I'm just going to skip this one in the interest of time. And we see when we house these mice in just a quiet satellite facility where there's not a lot of extraneous noise, we do get some loss of the efferent, the medial efferent synapses contacting these outer hair cells. It's primarily in the lower frequency regions and in the high frequency regions, and not so much in the regions of best sensitivity. When we look in the brainstem, something that is much harder to do, again, with human tissue, so we stick with our mouse model here. We can count the cell bodies for these efferents labeled with cholinesyl-transferase. And what we see with age is that we lose medial efferent synapses, but not so much the lateral, medial efferent neurons, but not the laterals. And I don't have the data shown here, but I'll just say this seems to be a particular vulnerability of the medial efferent neurons because there was no change in the vestibular efferent neurons or the trigeminal motor nucleus neuron control. What's really interesting, though, is that this loss of medial olivocochlear neurons actually correlates with poorer sound detection and noise as measured using an ABR, but only in our older animals. So there was no correlation with response thresholds in quiet and no correlation in the younger animals, only the older animals. So this loss of medial olivocochlear neurons with age seems to be really mostly a problem for aging animals, possibly because they don't have other compensatory mechanisms or weakening of compensatory mechanisms that normally could step in to help mitigate this loss of neurons and this poor hearing and noise. So, you know, ABRs are great. They're super convenient in mice, but they obviously don't reflect an awake, behaving, perceiving organism. So we wanted to know how this loss of the medial olivocochlear neurons with age may correlate with behavioral hearing deficits so that we can try to bring this back around to more of a clinically relevant situation. You don't typically have patients coming to the clinic with a complaint that they have an abnormal ABR. They come to the clinic because they are having behavioral problems, hearing and noise, right? So we can do this in mice. It's really a lot of work to track them with age behaviorally, but in collaboration with Michelle Dent's lab at the University at Buffalo, we have developed an assay for doing this. So the mice are trained to, in this case, detect an ultrasonic vocalization, which is their natural vocalization in quiet or noise, and they were tracked for 3 years by a heroic effort by Anastasia Kobrina, the graduate student in that lab, in the Dent lab. And you can see what happens is in about midlife here, 600 days is about midlife for a mouse, a CBA mouse, they rapidly start to lose their sensitivity for these vocalizations. And that starts to happen a bit earlier in noise than it does in quiet. And this coincides with this loss of medial olivocochlear neurons. However, the situation is a little bit complicated because the only thing happening is not just the loss of the medial efferent neurons. We're also, of course, losing hair cells, and we're losing auditory nerve ribbon synapses in these aging animals. And of course, there are other problems happening with the ear with age, stria vascularis, degeneration, etc., and maybe also other problems with the brainstem. So this makes it difficult to very specifically point to the olivocochlear loss to this hearing, as a contributor to the hearing and noise deficit, but we're pretty sure at least it's certainly not helping their ability to hear a noise, that they're losing these olivocochlear efferents, which normally would suppress the noise activity. Right. So to make the situation just a little bit more complicated, we're not just losing things in the cochlea. With age, it turns out we are actually gaining some efferent synapses on the inner hair cells. So this is a transmission electron micrograph cross-section through the base of an inner hair cell. And we did this project with Howard Francis and Dave Ryugo as in response to some surprising observations that Howard Francis was making when he was really characterizing the changes to the afferent synapses that occur before loss of the spiral ganglion cell body with age-related hearing loss. So basically what you see here in the young animals, the situation is that you have a lot of afferent synapses. Here's a nice ribbon synapse with the inner hair cell, and you have these small lateral efferent synapses that are formed with those afferent synapses, but not with the inner hair cell itself. But in aged animals, here, you see a cross-section through an outer hair cell. First of all, the outer hair cell is kind of shrivelly and not super healthy, not really a surprise with an aging hair cell. Sorry, inner hair cell, not outer hair cell. But surprisingly, there were these huge efferent synapses formed directly with the inner hair cells here that we just did not see in the young animals. And this is over here just showing the 3D reconstructions of those, showing no efferent synapses directly with inner hair cells in the young, and huge ones here in the red in the old animals. And here's just the quantification of that. These are the direct synapses with the inner hair cells going way up in the old animals. Seems to be related to loss of afferent synapses. So when about half the synapses are lost is when we see this re-innervation of the efferent of the inner hair cell with these efferents. And it's really recapitulating a situation that occurs during development, which seems to be a bit of a surprise to us, but it is what it is. We aren't sure if those are medial efferents or if those are lateral efferents. There is a slight decrease in those exodendritic synapses directly with the type 1 spiral ganglion neurons, but it's not on par with the increase in direct innervation of the inner hair cells we see. But it doesn't really matter because the net effect is that whichever kind of efferent it is innervating the inner hair cells directly, it actually has an inhibitory effect on the inner hair cell activity. So this could be good because it may be protecting against further ribbon synapse damage, further damage to the inner hair cell from lots of activity, but it's going to suppress the auditory nerve response to a sound, which means that's going to exacerbate the hearing loss further. So this is all super interesting that we see this in these animals, in these mice, in these mouse models, but I guess the question is this relevant to anything outside of a mouse that lives in a laboratory, right? And I'll tell you, we'd really like to look in human ears with more well-characterized hearing function and noise exposure histories, but the logistics of that are really, really complicated and very expensive and very difficult. So at present, while we try to figure out a good way to do that in human ears and human brainstems, we're starting to turn to other species that actually naturally live with a lot of noise in their lives. Okay, so in this case, we're looking at bats with Cynthia Moss's lab. So if you're not familiar with bats, they use echolocation to survive, basically, to forage for food, to navigate around. And different species of bats have different echolocation behaviors and different social behaviors. So some bats are, in these huge roosts that are extremely noisy, and their flight behaviors when they're echolocating are among many other bats, echolocating at about 140 decibels. So these animals are producing these ridiculously loud sounds and receiving the echoes back from them, and they're also exposed to other bats producing these really loud sounds all the time. But you can imagine if they lose their hearing, they're not going to do very well out in the wild because a deaf bat can't eat. And so we're looking at the efferent specializations in these animals. And surprisingly, there's little information in the literature. There's one species of bat, the mustache bat. There's a little bit of data showing they have single, very, very large olivocochlear efferent neurons contacting the outer hair cells, and that they have a large number of efferent neurons based on their size and species. So there's one species that has this. There's another species that doesn't have any medial olivocochlear efferent neurons at all, only laterals. We are particularly interested in the big brown bat, which is a native North American species that has very well characterized echolocation behavior, but also has recently been shown to actually be resistant to intentional acute noise exposures. So what we think is that these efferents are providing more inhibition to the outer hair cells. Maybe there are also more inhibition to the inner hair cells in response to loud sound exposures. And this not only helps to protect their ears against damage accrued over time, probably also helps them attend to their own vocalizations and tune out vocalizations that they don't want to listen to, tune out the chatter, tune out the noise. So we're still working to quantify this data. It's actually almost as difficult to procure and deal with bat inner ear tissue as it is to deal with human inner ear tissue. But we do, we do what we can to harvest tissue from animals that are at the end of their experiments, basically for physiological experiments and other purposes. We figured out a way to determine the age of the bats based on their DNA methylation in collaboration with a lab at University of Maryland. And I hope in the next year, we really will have a better handle on the specializations in these species that are evolved to live in very noisy environments with a lot of exposures. All right, so I'll try to wrap up and leave a little time for questions. What do I want you to know? I just showed you a ton of data. I told you a lot of information. Hopefully you didn't all tune out. I just want you to know it's complicated, really, to understand the role of the olivocochlear efferent system in hearing, largely because the system is interacting with acoustic experience, and with age. There are age-related changes. There's plasticity. And this is all occurring alongside all of the well-known afferent changes that occur in the auditory system with noise and with age. What haven't I shown you? There's a lot to do in this system to try to tease out its role in hearing. So for instance, we want to know really how is this system involved in processing more biologically relevant acoustic conditions. Most of the existing data are on tones in noise, and those are very convenient for measuring physiological responses, but less relevant to what we're all listening to on a daily basis. So we would like to try to develop assays for testing the olivocochlear system activation or suppression, and how that affects listening to more complex sounds like vocalizations and noise. We also haven't really tapped into the attentional effects. All those ABR measurements I showed you, the animals are anesthetized, they're not attending at all. And the behavioral tests they are attending, all of these things may actually affect how the olivocochlear effects are manifest, in addition to things like practice and learning. I already mentioned we want to look at bats. We really want to look also in general at non-inbred species. laboratory mice are very convenient, but they're all kind of the same. And so we want to know what is the diversity of the olivocochlear changes with noise and with age, and how does that relate to the heterogeneity we see in humans. So I'll refer you to a recent review we published if you want to know more about the behavioral effects of the olivocochlear system and other conditions in which that system is abnormal or affected. And I just would like to thank my lab for really being excellent people to work with. They're great scientists. They're really productive. They're fun to have around. I'm really lucky to have them working with me. I have great collaborators, and we have some wonderful support staff at Hopkins for working on this question. And of course, I need to thank the funders for paying us to do all of this fun work.